So we are going to do chromatography, and we've done chromatography before on plant pigments. This time we're going to do chromatography on animal pigments. So pigments, remember, are light absorbing molecules, and animals have pigments in their eyes, so it helps them to absorb light and perceive information about the world around them. So I have a piece of chromatography paper set up here, and I've got a couple of tick marks. This one's labeled wild. This one has a B for brown. This one has an S for sepia. This one has an R for rosy, WH for white. So those are all different phenotypes of fruit flies. The wild type fly makes all the pigments, so that's the typical type fly. The brown, the sepia, the rosy, and the white are all mutants. Remember we talked about Thomas Hunt Morgan. He was the famous geneticist who studied fruit flies. These mutants were super important to his research, and today in this activity we're going to use these mutants to try to understand something about the relationship between proteins and phenotypes. And the proteins that we're going to be looking at are these pigment molecules. So the kind of weird thing that we have to do in this lab is apply the fly pigments to the paper in order to do chromatography. So I am going to smash the heads of the flies on these little dots. So right now I'm smashing the wild type flies on that little tick mark that I showed you for wild type. And I'm going to do 4 of each of the fly phenotypes. And I'm trying to smash just their heads where their eyes are, but I'm really smashing a lot of their bodies. So this is my 4th one. So basically, I'm just trying to get a big clump. I'm just trying to get a big clump of eye pigments from this fly on the paper. Let's move this guy back over here. Okay, so hopefully we got some pigments there. I'm going to switch the rods and move on to the brown. So I don't want to get any of those wild type pigments onto the brown tick mark. So I switched my rod, and now I'm going to smash these guys' heads, 4 of them, on the brown spot. It's hard to just get their heads, but I'm doing the best I can. Here's the 3rd one, and a 4th one. Okay, so now I'll grab a new rod and we'll try the sepia. So I'm going to smash 4 of these guys on my sepia tick mark. Just trying to get a bunch of this eye pigment on this spot. And then just like we did with the plant pigments, we'll use a chromatography solvent to try to separate out the different molecular components of the eye pigments. We'll see which of these flies has or does not have certain pigments. We'll do 1 more of these guys, and then we'll move on to the rosy. Okay, grab a new clean rod. Actually, we'll put these guys here. Grab the rosy, and we'll do 4 of these guys. Kind of hard to smash. that guy by the head. And here's our last one. Okay, and then finally, we'll put the rosy back, and then we'll do the white. So these guys, generally, what I think we're going to find here is that they're probably not making any pigments, which is why they appear white. We'll have to wait and see. There's one. Here's the second. And a third. And a fourth. Okay, so now at this point, we should have a cluster of pigments associated with each of these phenotypes. We're going to run them using an ammonia-based chromatography solvent for about an hour, and then we'll look at our results. The results should tell us which pigments were present for each phenotype.